Hello students, welcome to Read MedPrep Academy channel. Today in Sexual Reproduction in Plants Part 3, we are going to talk about a pre-fertilization structure, the gynoecium, which is the female reproductive part. The gynoecium represents the female reproductive part of the flower. The word gynoecium represents one or more pistils of a flower. What is a pistil? The word pistil refers to the ovary, the style and the stigma. A pistil is derived from a carpel. Ovary represents the part that contains the ovules. The basal swollen part of the pistil is the ovary. The ovules are present inside the ovary cavity, which is called the locule on the placenta. Here you can see the ovary and the cut section of the ovary showing the locule, and the ovules are arranged over one another. What is stigma? Stigma serves as a landing platform for the pollen grains. What is style? Style is an elongated slender part beneath the stigma. Gynoecium or the carpel arises as a small papillate outgrowth of the meristematic tissue from the growing tip of the growing primordium. It grows actively and soon gets differentiated into ovary, style and stigma. How many ovules are present? The ovules or the Megasporangia arise from the placenta. The number of ovules may vary in an ovary, maybe one like in the paddy, wheat and mango, or many ovules may be present in the ovary like the papaya, watermelon and orchids. Here you can see a single ovule attached to the placenta on the left side and the multiple ovules in the locules present on the right side. Now what is the structure of the ovule or the Megasporangium? Ovule is also called Megasporangium and is protected by one or two coverings called integuments. A mature ovule consists of a stalk and a body. The stalk or the funiculus, also called funicle, is present at the base and it attaches the ovule to the placenta. The point of attachment of the funicle to the body of the ovule is known as the hilum. It represents the junction between the ovule and the funicle, which you can see the structure on the right side. What is a raphe? In an inverted ovule, the funicle is adnate to the body of the ovule. It forms a ridge called the raphe. What is nucellus? The body of the ovule is made up of a central mass of parenchymatous tissue called the nucellus, which has large reserve food materials. The nucellus is enveloped by one or two protective coverings called the integuments. The ovule with one or two integuments are said to be unitegmic or bitegmic ovules respectively. You can see a bitegmic ovule which has two layers of integuments on the left side and a single layer of integument on the right side. What is micropyle? The integument encloses the nucellus completely except at the top where it is free and forms a pore called the micropyle. What is chalaza? The basal region of the body of the ovule where the nucellus, the integument and the funicle meet or merge is called the chalaza. What is an embryo sac? There is a large oval sac-like structure in the nucellus which is present towards the micropylar end. This is called the embryo sac or the female gametophyte. It develops from the functional megaspore formed within the nucellus. On the right side you can see the embryo sac that is present near the micropylar end, the egg cell and the two synergids. The integumentary tapetum. In some species, otherwise the unitegmic nucellate, the inner layer of the integument may become specialized to perform the nutritive function of the embryo sac, and this is called the endothelium or the integumentary tapetum. The example of such type of integumentary tapetum is found in Asteraceae. What are the types of ovules? There are two types of ovule based on the position of the sporogenous cell, tenuinucellate and crassinucellate. What is tenuinucellate? If the sporogenous cell is hypodermal with a single layer of the nucellar tissue around it, this is called tenuinucellate. Normally, tenuinucellate ovules have very small nucellus. What is crassinucellate? Ovules with hypodermal sporogenous cell with multiple layers of the nucellar tissue around, and this is called crassinucellate. Normally, These ovules have a fairly large nucellus. What is hyostase? Group of cells that are found at the base of the ovule between the chalaza and the embryo sac is called the hyostase. What is epistase? And the thick-walled cells that are found above the micropylar end above the embryo sac is called the epistase. Now here you can see the structure of the ovule, where it has an integument, it has a funicle that carries the vascular supply which attaches the ovule to the placenta, and the attachment of the funicle to the ovule is called the hilum, and it has a thick ridge called the raphe, and the integument is open at a place that is called the micropyle. Inside the integument is the nucellus which contains the embryo sac. Opposite to the micropylar end is the callosal end. You must study the structure of the ovule properly. The classification of ovules. Ovules are classified into 6 main types based on the orientation, form and position of the micropyle with respect to the funicle and the chalaza. What are the types of ovules? Orthotropous, anatropous, hemianatropous, campylotropous, amphitropous and succinotropous. Here you can see in the images the different types of ovules. A is orthotropous, B is anatropous, C is hemianatropous, D is campylotropous, E is amphitropous and F is succinotropous. Let us discuss about these ovules one by one. What is orthotropous ovule? In this type of ovule, the micropyle is at the distal end, and the micropyle, the funicle and the chalaza, everything lies in a straight line. Example is Piperaceae and Polygonaceae. Here you can see in the image the micropyle, the embryo sac and the chalaza and the funicle are in a straight line. What is anatropous or inverted ovule? The body of the ovule becomes completely inverted so that the micropyle and the funiculus come to lie very close to each other. This is the common type of ovules found in dicots and monocots. In the image, you can see that how the anatropous ovule appear. What is hemianatropous ovule? In this, the body of the ovule is placed transversely and at right angles to the funicle. Example is Primulaceae. You can see in the image the micropyle, the embryo sac, the chalaza being at right angles to that of the funicle. What is campylotropous ovule? The body of the ovule at the micropylar end is curved or more or less bean shaped. The embryo sac is slightly curved. All the three, the hilum, the micropyle and the chalaza, are adjacent to one another, with the micropyle oriented towards the placenta. Example of this campylotropous ovule is Leguminosae. Here you can see on the right side the bean-shaped structure of the ovule. What is amphitropous? The distance between the hilum and the chalaza is less. The curvature of the ovule leads to a horseshoe nucellus. Example is Erismataceae. Here you can see in the image the horseshoe-shaped ovule that is present. What is succinotropous? In this, the funiculus is very long and it surrounds the entire ovule. Example is Cactaceae. Now let us move on to Megasporogenesis. What is Megasporogenesis? The process of development of a megaspore from a megaspore mother cell is called Megasporogenesis. What are the steps involved in the formation of Megasporogenesis? Formation of the archisporium, division of the archisporium, division of the parietal cell, division of the megaspore mother cell and formation of a functional megaspore. Now let us discuss one by one. The formation of archisporium. As the ovule develops, a single hypodermal cell in the nucellus becomes enlarged and it functions as the archisporium. In some plants, the archisporial cell may directly function as the megaspore mother cell. Next step is the division of the archisporium. In some plants, it may undergo a transverse division to form outer primary parietal cell and an inner primary sporogenous cell. Now the third step is the division of the parietal cell. The parietal cell may remain undivided or sometimes divide by a few periclinical and anticlinical divisions to embed the primary sporogenous cell deep into the nucellus. The stage number 4 is the division of the megaspore mother cell. The primary sporogenous cell functions as the megaspore mother cell. The megaspore mother cell undergoes meiotic division to form 4 haploid megaspores. This is called megaspore tetrad. The fifth stage is the formation of the functional megaspore. Out of the 4 haploid megaspore tetrad, 3 megaspore degenerate and one of them develops into a functional megaspore. Here in this image, you can see the various stages of Megasporogenesis. A is where the primary archisporial cell is formed in the sporogenous tissue. B is where the archisporial cell divides into 2 cells, the primary parietal cell outer side and the primary sporogenous cell in the inner side. And stage number C is the differentiation of the primary sporogenous cell into the megaspore mother cell. Stage number D is the formation of the megaspore mother cell to linear tetrads or tetrad megaspores. And stage number E, where the 3 megaspore cells degenerate and only 1 megaspore becomes functional and becomes the ovule cell. The types of megaspore development, based on the number of megaspores that develop into the embryo sac, there are 3 basic types of development. One is called monosporic, the other is bisporic, and the third one is the tetrasporic. What is monosporic? Here, the functional megaspore forms the female gametophyte or the embryo sac. Since only one cell directly forms the ovule or embryo sac, this is called monosporic development. Example, polygonium. Here you can see in the image, the upper image shows that the 3 megaspore mother cells degenerate. Only the functional cell forms the ovule or embryo sac, and in this, when it undergoes a mitotic division, it forms 2 cells, and when 2 mitotic divisions occur, it forms 4 cells. In the image on the right side, you can see that the monosporic cell undergoes trimitotic division, resulting in the formation of 8 cells. What is bisporic? Of the 4 megaspores formed in the tetrad, 2 are involved in the embryo sac formation, and this development is called a bisporic. Example is allium, and the 2 cells undergo bimitotic division to form 8 nuclei cells. What is tetrasporic? All the 4 megaspores are involved in the formation of the embryo. This is called tetrasporic. That means there is no degeneration of any of the megaspores. Example, peperomia. Here you can see in the first image on the left side, a tetrasporic cell undergoing a bimitotic division, resulting in the formation of 16 cells. On the right side, you can see a tetrasporic cell undergoing a bimitotic division and forming 8 cells. Now, let us discuss about the monosporic embryo sac. An ovule generally has a single embryo sac and is called monosporic embryo sac. Example is polygonum type. Here you can see the development of the ovule and the embryo sac of the polygonum type, where the nucellus forms the archisporial cell. The archisporial cell develops into a megaspore mother cell. The megaspore mother cells forms a megaspore tetrads, and one of the megaspore becomes a functional cell, and this functional cell, which has a single nucleus, divides and forms the 2 nucleate stage, and again it undergoes mitosis to form the 4 nucleate stage, and this again multiplies and forms the 8 nucleate stage, which is the complete ovule. Now, what are the stages in the development of the formation of monosporic embryo sac? One is the elongation of the megaspore. Number 2 is the division of the nucleus, resulting in the formation of a binucleate cell. Number 3 is vacuolation. Number 4 is 4 nucleate stage, Number 5 is 8 nucleus stage, and number 6 is the organization of the embryo sac. Now let us see one by one. Elongation of the megaspore. We know that the single megaspore is functional and is the first cell of the embryo sac or the female gametophyte. The megaspore elongates, develops, expands along the micropylar and the callosal axis, and the other three degenerate. And the division of the nucleus occurs. The nucleus undergoes a mitotic division resulting in the formation of binucleate cell. In this stage, there is no wall formation. Third stage, vacuolation. A central large vacuole appears between the nuclei, and the vacuole slowly expands and pushes the nuclei to the opposite poles of the embryo sac. Now, fourth stage is the 4 nuclei stage, where both the nuclei divide mitotically and there is a central vacuole. On either side, there are 2 nuclei. In the fifth stage, otherwise called the 8 nuclei stage, the 4 nuclei divide mitotically, forming 8 nuclei stage, with 4 nuclei that are present at each pole, 4 nuclei at the callosal pole and 4 nuclei at the micropylar pole. At this stage, all the 8 nuclei are present in a common cytoplasm with the vacuole. Now, let us move on to the organization of the embryo sac. After the last nuclear division, the cell undergoes appreciable elongation, assuming a sac-like appearance. This is followed by a cellular organization of the embryo sac. Now, what happens in the organization? The micropylar end nuclei, which are 4 in number at the micropylar end of the embryo sac, the 3 organize themselves into an egg apparatus, and the 4th one is left free in the cytoplasm of the central cell as the upper pole nucleus. There are another 4 nuclei at the callosal end, out of which the 3 nuclei of the callosal end form the 3 antipodal cells, whereas the 4th one that is present functions as the lower polar nucleus. Depending upon the plant, now there are 2 polar nuclei. They may remain free or sometimes they may fuse to form a secondary nucleus, causing a central cell. The egg apparatus and the upper pole nucleus form the micropylar nuclei. The antipodal cells and the lower polar nuclei form the callosal nuclei. Fusion of the polar nuclei may sometimes occur to form the secondary nucleus. In this image, you can understand better where the sporogenous tissue, which has a functional megaspore developed from the megaspore mother cell, has a single nucleus. It multiplies and forms a binucleate structure, then a 4 nucleate structure, then an 8 nucleate structure. The 4 nuclei are present at the callosal end and 4 nuclei are present at the micropylar end. At the micropylar end, one cell forms the X cell, and there are 2 synergid cells on either side, and 1 nuclei forms the polar nuclei. At the callosal end, the 3 nuclei form the antipodal cells, and the polar nuclei is in the center and joins with the another polar nuclei. What about the egg apparatus? The egg apparatus is made up of a central egg cell and 2 synergids, one on each side of the egg cell. What is the function of the synergids? They secrete chemotropic substances that help to attract the pollen tube. The special cellular thickening occurs called the filiform apparatus of the synergids, which helps in the absorption, conduction of nutrients from the nucellus to the embryo sac. And this filiform apparatus of the synergids also guides the pollen tube into the egg. Here you can see a mature embryo sac. At the micropylar end are the synergids with the filiform apparatus and an egg cell, and the central cell having the secondary nuclei called the polar nuclei, and at the callosal end there are 3 cells called the antipodal cells. So the entire mature embryo sac is a 7-cell structure with 8 nuclei. So today in sexual reproduction in plants, part 3, we discussed about a pre-fertilization structure, the gynoecium, which is the female reproductive part. Thank you. Kindly subscribe, like, share and comment to channel Read Med Prep Academy. Kindly log on to www.readmedprepacademy.com. Our Facebook ID is Read Med Prep Academy. Our email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.